Hello viewers, so this is the part 2 of chapter cell division. Part 1 was already uploaded. You can go and just watch that out to understand about the basics of chapter number 2, cell division of class 10 bio. So today I'm going to start with mitosis. So first of all, mitosis is a kind of cell division that will occur in all the cells in our all body part except germ cell. So it is going to occur in all the cells in our body except the germ cell. And in mitosis, one cell divides into two cells, right? So it means if this cell is going to divide in these two, so it is important that the content which was present here in the cell, it should be equally divided. Then only the cells would survive. And the new cell that we get from this division is said to be daughter cells. Daughter cells have nothing to do with male and female. It's just the name given to the cells, new cells that arises from the old cell. Now after this, in mitosis we have two things. First is in cell division. Basically in cell division we have two phases. One is interphase and the second phase is mitotic phase. And in mitotic phase we have four subphases here, which how you can learn PMAT, you can remember PMAT. P means prophase, M means metaphase, A means anaphase, and T means telophase. So here we are going to study each one of them separately. So first is the interphase, which is given here in your book on page number 14. Okay, so here in this part, prophase, metaphase, anaphase and telophase, we are going to study with first of all interphase. Interphase is also said to be resting phase. Why? Because in interphase it seems like there is no change that is taking place inside the cell, especially the nucleus. But in reality there are number of changes that a cell undergo. First of all, more synthesis of DNA takes place, more synthesis of RNA takes place, and the organelle like mitochondria and chloroplast, which have got their own DNA, they also get multiplied. And also along with that, as soon as this phase ends, so before that phase starts, you should know one thing. I mean, before that mitosis start, interphase is divided in three parts. So here are the name of the phases which is given in the book here. That first is your first growth phase. Very difficult to zoom in, I guess. First is the first growth phase, then synthesis phase, and then last is second growth phase. So you need to remember the sequence in which phase what will happen. So in first growth phase, RNA and proteins would be synthesized, because as I told you, the cell content has to be divided in two parts. So it has to be first of all made it to be double, so that half and half it goes, so again the number of chromosomes are maintained. Okay, so here RNA and protein will be synthesized more, and as I told you, mitochondria and this thing chloroplast, which have got their own DNA, they will also get multiplied so that they could be transferred further. And after that would be your synthesis phase. In synthesis phase, more synthesis of DNA will take place. Along with that, chromosomes will get duplicated. Duplicated means if there one chromosome is there, suppose I am giving you an example, there is not one chromosome, one is there, it will turn into two. And the last is the second growth phase. In second growth phase, more of RNA and protein would be formed. So in first, RNA and protein, and if any cell organelle like mitochondria and chloroplast having their own DNA, they will get multiplied. Then in synthesis S phase, DNA only, and in growth phase, more amount of RNA would be formed. And this interphase is actually, very long phase for a cell, because most of the time cells remain in this phase only. And this mitosis, which comes after G2 phase, it is quite quick phase for the cell, quite short phase compared to that of interphase. And so there is a summary given here, so you can read what all changes occur in a cell, what all things get multiplied under which phase. Now after G2 phase, that is second growth phase, now there is next phase, that is mitosis. Mitosis, I told you there would be four subcategories here, four phases that would be taking place before the nucleus and cytoplasm they finally divide. So first would be the prophase. Okay, if you see, it's given here the diagram. Here in this diagram you can see only chromatin fibers are visible, right? Nucleus is well defined and centrosomes are there, right, which is shown here on the top. Now when the resting phase is done, now it enters the prophase. All these phases are very important because one or the other year they do ask. So like last year they have asked about anaphase, they had asked. So maybe this year they might ask you to draw, you know, that particular phase, you never know. So it's always better to remember all these phases which are given under mitosis. So under prophase we have two types, early and late. See, in exam you can write simple prophase also. If you want to be very specific, then you can write early or late prophase, right? Okay, so here what can you see in this diagram? Now chromatin fibers have changed into chromosomes. You can see well defined chromosomes are here, right? Along with that, see here the centrosome was only one, so it has duplicated itself. Two have been formed. One will be moving here and one will be moving here in the late prophase. So you can see in the next diagram, if you can see, what is the difference between these two? First of all, here the centrioles were there on the top. Now in this diagram it has come here at the corners, and also nucleus was well defined here, and here it has disappeared. And also this centriole is going to give rise to spindle fibers. Spindle fibers are nothing but thread like structures which are going to pull these chromosomes apart. They are going to separate them and they are going to pull them apart so that they are divided equally. Okay, so here if you see, similar diagram is given for plant, but make sure in plant there will be no centriole, right? You can see the difference. Everything is same, there is no centriole, right? Okay, after the prophase all these things has happened. What has happened? First of all, your under this prophase you are able to see chromosomes, they are very distinct. And second thing, centrioles have been duplicated, and after, like by the end of this prophase, they are going to move at this position, equatorial position basically. And after that they are going to, there would be removal of nucleus as well. After that is metaphase. So in metaphase, if you can see, the chromosomes have all lined up in a straight line, kind of a straight line, and this we say is a equatorial plane. See, we know equator, like we study in geography, equator is said to be, you know, horizontally and all, but here equatorial is this, in a straight line, right? And that is the main feature of metaphase, that these chromosomes, they will align themselves in one of the planes in equator. Same will happen with plant as well, just the difference would be there would be no centrioles. Okay, next is anaphase. If you can see here, they had sister chromatid, there was double na. Okay, if you can see here, yeah, that is better I think. So here you can see there are two sister chromatids in each, but when you come to anaphase, in anaphase they have been separated. And which thing has done that? This spindle fiber, I told you, web like structure would be generated which are going to pull chromosomes apart. So here this web like structure is going to pull these chromosomes apart, and you can see they have separated. So one pulling from this side, one pulling from this side, so they will separate in anaphase. And also by the end of anaphase, see, they have given early and late. By the end of anaphase you will see a furrow. Furrow means a little, you know, division kind of thing you are going to see here. And in plants you will see there is no furrow, of course there would be cell plate which is going to come in between, we will see that. Okay, now by the end you can see here carefully, these chromosomes which were here almost at the center, they have gone to the corners and furrow has been developed. So means cell is going to divide from here. Okay, now after that what will happen? In telophase mein what will happen? These spindle fibers, they will vanish which was coming out of centrioles, and you can see that. And also along with that, nucleus will again reappear, spindle fibers will disappear as I said, and chromatin threads now would be seen. Chromosome will disappear because they have been divided in two nucleus. And also here, if you can see, in plants we'll have cell plate instead of furrow. Furrow is this from where the cell is going to divide, and by the end you are going to get two different cells that are called daughter cells. And because here first of all the nucleus has divided and then cytoplasm has been divided accordingly, right? So we have two words that we have to remember here. One is cytokinins. Cytokinins means where the, here it is given, where the cytoplasm divides, means division of cytoplasm is said to be cytokinesis. And one is karyokinesis, where the nucleus divides. So in this process that we had studied, so here two things had happened. Firstly, the nucleus is divided, and then after that the cytoplasm gets divided according to the division. And since you can see from here, we had started from this cell, single cell here, this one, and ultimately we got two different cells, right? And the content has been divided equally into both of them. And in case of plant, you will have a cell plate which is going to be almost at the center, and it is going to divide two cells completely, right? So again, in mitosis, it is a kind of cell division where one cell divides into two, and for that to happen, it is important that the content present in the cell should be divided equally among both the cells. So for that, what happened, first phase that we have is interphase, which is said to be resting phase. Why resting? Because it seems like cell is not doing anything, but in reality lot of reactions are taking place inside the nucleus, that is multiplication of DNA and our RNA, proteins and all is being divided. After that is being done, and I explained that there are three phases, right, which I mean the three phases in interphase. One was G1 phase, then was S phase, and other was G2 phase. After G2 phase, the cell can enter, I mean will of course enter into the next phase, that is mitosis. And under mitosis we have four different phases: prophase, metaphase, anaphase and telophase. Okay, so suppose in exam you get a question. Let me give you one example here, the question that we got in board exam. Okay, so this is this year's paper, bio paper, and the question that we got this year was diagram based, the last question here, this one, if you can see, right? As I told you, diagrams from this chapter is quite important, so you cannot skip that part. This topic is itself is very important. Okay, so the diagram states that you need to identify the given stage. So as you can see, chromosomes, they have already separated and they are being separated, right? But there is no furrow. But you can see under this thing, that is under anaphase, we have got two phases now, one is this, one is this. So you have to write anaphase, because under anaphase both these things come, like chromosomes being separated and they will be moving apart and furrow is developed. So this would be, they are saying identify the stage. So stage is anaphase, right? You will not write late anaphase or early anaphase. No, you have to write anaphase because they are asking you the phase, right? After that, they are saying give one reason to support your answer. So why? Because you will say the chromosomes, the sister chromatids, they have been separated, into two. So that is why, and they will be moving towards the centrioles, right? So, or if you want to write exactly, you will write about this, that centromere attaching two chromatid divides or split, sister chromatids of each separate are drawn apart towards the opposite pole. So this you can write. And the number of chromosomes given in the diagram. So we know two sister chromatid make one chromosome, 1, 2, 3, 4. So your answer would be 4 chromosomes. So like this you have to do this topic.